everyone, today I'm going to talk to you about this poster which you see in front of you, which discusses the genetic diversity of the critically endangered Lake Minnow in Poland and its implications for conservation in the future. The main aim of this study was to investigate the genetic diversity of the Lake Minnow, a tiny endangered cyprinid fish found in Poland and other countries in the Northern Hemisphere, and for whom more genetic information is required in order to put in place a more effective protection strategy. To carry out this study, they used microsatellite polymorphisms to characterise the genetic properties of the Lake Minnow, and they sampled from 12 populations across Poland, as you can see here in this map in Figure 1. Adult fish were captured from each population using baited traps, and samples were taken from each of their left pelvic fins before the fish were returned to the water from where they were caught. The samples were then preserved using an air dryer. DNA was extracted from these samples using an extraction and purification kit, and these extractions were then stored at -20°C until it was time for PCR amplification. The PCR amplification that was carried out used primer sequences from previously published studies and from GenBank. The PCR cycle itself began with the degeneration of DNA at 95°C for 3 minutes, and then, as you can see here, there were 34 cycles, beginning with denaturation for 1 minute at 95°C, annealing for 35 to 40 seconds at 52 to 65°C, and then 35 to 45 seconds of elongation at 72°C. After the final cycle, there was one last elongation which lasted 15 minutes at 72°C. After PCR amplification, the PCR product was then verified using electrophoresis in a 2% agarose gel and stained with ethidium bromide. After the verification of the PCR products, they were sent off for sequencing, and upon receiving the results from sequencing, statistical analyses were then carried out. Among these analyses was the exact Hardy-Weinberg test, which tests for deviation from the Hardy-Weinberg equilibrium. The occurrence and/or influence of the founder bottleneck effects was based on the Garza-Williamson index, and the genetic divergence between the population was calculated by looking at the FST values. The results of the analyses showed that across all of the investigated markers, there were 96 alleles detected, with the allelic frequency being low and varied. The results of the Hardy-Weinberg test show that across all markers, none of the departures from the Hardy-Weinberg equilibrium were significant. Meanwhile, the average Garza-Williamson index indicated that the founder and/or bottleneck effects had an impact on genetic variation between all of the populations. Finally, based on the FST values, all the genetic distances between the populations were found to be very large, and the genetic differences between the populations were all found to be significant. So, what do these results mean in terms of the implications on active protection? We already know that active protection programs are designed to preserve genetic differences between populations. The current active protection programs for the minnow in Poland are currently limited to Warsaw, with programs including captive breeding and the translocation of cultivated fish, which stimulate gene flow on a local scale. The methods that were developed and applied in this study have the potential to help maintain the genetic variation of the lake minnow populations. The molecular markers that were used in this study enable the identification of males and females in the population who are most genetically different to each other. This means that they can assemble spawning pairs and produce genetically diverse juveniles. Another method of conserving genetic variation could be to create a cryopreserved sperm bank. This would be constructed by using the genetic profiling of males in the lake minnow populations across Poland. This method would be especially valuable in the conservation of populations that are more isolated or are more fragile to environmental change. Thank you for watching my presentation.